thank you. I wanted to start, we all do in optics and we all like to focus in, but people who do optics know that if you want to focus really tightly, you have to start big. So let's start with probably one of the biggest problems you can think about, or the most interesting, to me at least. And also Schrodinger thought about this, is this idea of what is life, or how does it work? And I remember seeing this video on a TED lecture by Drew Barry, and it shows the replication of DNA. And what you notice is that these proteins are really behaving like machines, like mechanical machines you might expect to see in a factory, essentially taking apart, unzipping this DNA, and then creating copies of it. It's amazing the complexity and the order that's involved. And this is really what Schrodinger was talking about. He basically said that whereas physics is trying to make everything disordered, through entropy, what life has done is invented this molecule, or he's talking about proteins, that have extreme order, and this extreme order is really at the heart of what life is about. And to understand how these proteins work, we want to understand their mechanical motion, how they move, okay? So this is a protein called albumin. It has two different vibrational motions are shown here, okay? So I'm going to talk about how we can access those types of motions with light today. Okay, so this is in the area of optomechanics, and specifically nano-optomechanics. Now in the previous talk we saw that there was a relationship between size and the frequencies that were involved, and in the previous talk we heard that you could go to sort of picosecond type timescales and even beyond that. And so these are really the types of timescales that you need. If you're talking about nanoscale objects, relatively big nanoscale, like 100 nanometer type objects, then you get these optical resonators that are also mechanical resonators, and their mass is around here, and the frequency they have is around 5 gigahertz, okay? So you can imagine doing experiments with these directly. But now, if you're talking about the oscillations of a protein, the mass is much smaller, and the frequency modes actually are around hundreds of gigahertz into the terahertz range, up to several terahertz. So you really have to come up with new tools if you wanted to actually probe individual proteins and look at how they're behaving, okay? Now, these vibrational modes, everything has them. At an even smaller scale than proteins is C60. This is carbon buckyball. Essentially, what are shown here are two acoustic modes of this C60 molecule. It looks like a soccer ball. And you see you have the L equals 2 mode, which is a sort of an accordion-like mode, and I've shown the deformation that you would get for that. And you have the L equals 0 mode over here as well. And these two modes have frequencies that are in the terahertz range. Okay, so this object is about 0.7 nanometers across, and you have to go into the terahertz range frequencies. And this type of, these vibrational resonances here, they really tell you about the large-scale oscillations of this C60 molecule. Whereas if you go into the higher wave number, 1,000 wave number or so regime, then you're really looking at the atomic interactions that are very local, okay? So the types of oscillations we're looking at are these types of global, larger scale oscillations, okay? So what I'm looking at is nano-bio, okay? So not quite as small as C60 over here, but looking at things that are in the single-digit nanometer range, which could be proteins, or proteins interacting with DNA, or DNA by itself, or even viruses, okay? So all of these things, I'm going to show you how we interact with them and study them with light, okay? So I guess one question you could ask yourself, if you could hear a protein or a particle, what would it sound like? What would its sound tell you about how they operate and how they behave? Okay, so that's really the type of motivating question that we have here. And these are the types of spectra that we get. I'm just showing you what I'm going to get to later on in the talk, sort of to whet your appetite. But essentially, these are two different proteins, and I'll show many different examples later, where you have very different spectra in the sort of sub 100 gigahertz range where we've scanned here. So this is in the single-digit wave number range as well. And you can see very distinct spectra for every single protein that we probe. As well, it's highly reproducible. And these are all single protein measurements. So we're looking at one protein at a time, okay? Okay, so we use a technique called optical tweezers, and there's an optical trap-in session in this conference, which I really like. And so what optical tweezers do is they use the momentum of light to manipulate small objects. So if you have a focused laser beam now, if a particle comes into the focus region, it's going to scatter photons away from the focus. And what this does is these photons have momentum, and so there's going to be a reaction force that's going to push the particle towards the focus then. And in this way, you can kind of pull particles towards the focus of a tightly focused laser beam. Okay, so this is sometimes modeled with a Hookean type of expression, and the force scales as the field squared, or the intensity of light, the gradient of that, actually. So the problem with this is it also depends on the polarizability of the molecule that you have, and the polarizability scales as the particle size to the third power. So there's this large, strong scaling on the size of the particle with the forces that you can obtain to do laser tweezers or optical trapping. Okay, so this is the Rayleigh scattering limit. So this limitation was recognized even in the first works looking at optical tweezers. For example, this work by Arthur Ashkin and co-workers, basically they're focusing a laser beam into a fluid and looking at the trapping of nanoparticles. And one thing that they realized right away is that you could get down to about 100 nanometers with a conventional laser tweezer, but then when you go below 100 nanometers, you essentially are blowing up the particle, or you're not able to trap it for long enough, because you have to use such high intensities to hold onto the particle that it's just not a practical solution. So we've come up with a solution to overcome that problem, and this solution is to use an aperture in a metal film. Now, an aperture in the metal film, we all know from Bette's aperture theory, has a very strong scaling on the size of the aperture and the wavelength as well, just like Rayleigh scattering, but this is actually a fourth power dependence on the transmission as you make the wavelength larger than the aperture, or as you make the aperture smaller. So you get this very strong drop in the transmission. Now, if you put a particle inside the aperture though, all of a sudden you've made this aperture bigger, okay? The wavelength of light inside a dielectric is smaller, so another way of thinking about it is that the aperture is bigger in this case. It's dielectrically loaded, okay? And if you have a bigger aperture, you're going to get all of a sudden a lot more transmission of light. And when you get all this extra transmission, you're going to be able to see the particle in the aperture very clearly. Even though this is usually a particle that would not scatter very much light, all of a sudden you put in the aperture, because it has such a strong influence on the transmission through the aperture, it's really easy to see this particle when it gets into the aperture. And so there's going to be a lot more transmission through the aperture, and this is going to create a force acting on the particle to pull it back into the aperture. And so what we're doing is we've created a trick, okay? So you have Rayleigh scattering, which has this third power polarizability dependence, and then you have this Bette's aperture theory, which has a fourth power scaling, and so we basically canceled the two out, or we've planed off the Bette's aperture theory against this Rayleigh scattering. And in doing this, we are able to get down to trapping very small objects in the single-digit nanometer range. So the experiment to do this is actually very simple. I've already showed you how we do it. You essentially just focus light onto a nano-aperture in a gold film and look at the transmission. And when you get a particle trapped, you should see a jump in the transmission. So you're just monitoring the transmission through a nano-aperture. Okay, it's very easy. And this is exactly what you see. For 100 nanometer particles, with less than a milliwatt of light, you get these jumps occurring. And notice that Ashkin was using 15 milliwatts of a shorter wavelength laser in his experiment. So this is very much, considering the third power scaling, this is very much more efficient for trapping small particles. And then so each of these jumps represents the trapping of a single 100 nanometer polystyrene sphere in an aperture. If you increase the power a little bit, then you're going to extend the trap-in time a little bit longer. And up with even less than a milliwatt, we can essentially trap this particle indefinitely. So we can hold on to it for as long as we want. Okay, and there's a distinct step, a big step, of the order of 20% in the transmission through this aperture. So it's not a subtle event. These are very easily detectable single nanoparticle events. Now I should mention that our chairman actually was looking at this stuff way back in 1999 and calculated 100 nanometer apertures and found that they could get good trap-in ability, but for 1,000 nanometer particles. So still relatively large particles that you can trap with conventional optical tweezers. And there were some experimental works, but again, they didn't really push it down to this 100 nanometer. They're looking at, or below, they're looking at this 200 nanometer range. But we want to look even smaller. We want to look in the range of a few nanometers, so looking at single proteins. And to do this, we wanted to change the shape of our aperture. So we use this double nano-hole optical tweezer for that. So double nano-hole is a nice shape. If you can make one nano-hole, you can probably make two. And then you just bring them closer together until the point where they just start to touch. And when they touch, the electromagnetic field gets focused at these cusps. And so you get this large light field enhancement there. And that's where you can get trap-in then. Okay, so we've been working on double nanoholes and related structures for quite some time now. So here's the experiment with the double nanohole. Just once again, this is actual experimental data. You have your particles diffusing around in solution. And at some point, one of the particles will get close enough to the trap, but the whole time you're shining a laser through the aperture, and this is showing kind of the noise on the intensity of light transmitted through the aperture. But when a particle gets trapped, all of a sudden there's a huge jump in the transmission. It's not subtle at all. But the other thing that's interesting about this is also the noise goes up by a lot. And this is not really noise. This is useful information. This tells you about the motion of the particle inside the aperture. So essentially, we can characterize our particles by looking at this, and I'll show you a lot more about that in the next couple of slides. Okay, so here's an example of trapping a single protein. This is bovine serum albumin. And we see initial trapping step here with a very small amount of noise in our actual system. This noise over here is actually telling us about the particle that's being trapped. So that's got useful information in it. So the signal to noise here is very large. As well, this protein actually has this sort of folding and unfolding motion, which we can observe when it steps down and then steps up and steps down. Okay, so you can actually observe protein folding at the single nanoparticle level, single protein level. Okay. Okay, so essentially we have a double nanohole. So we make two holes in a metal film, 100 nanometer gold film, or you can use different thicknesses here. And then we create a micro well in a cover slide, and then we put out a double nanohole. We first put our solution inside there that we want to analyze, and we put our double nanohole on top, and then we have our sample ready. And then we just put this in an inverted microscope setup, shine a laser in through the bottom, and this is a registration circle, but in the middle is this double nanohole that you can't see because it's quite small, but you can see some light being transmitted through it. So this is just to help us find the double nanoholes. So it's quite a simple experiment, as you can tell. So I want to talk about some of the interesting physical properties that we can probe with this. And here's an example where you could take a hairpin of single-stranded DNA. So if you have the complementary sequence for DNA on the second half of a single strand, it will hairpin on itself, and this will create this sort of double-stranded region here. This is 20 bases here, hairpinned around. Now, we can trap this single-stranded DNA. If we don't trap a hairpin, we just get the single jump. But if we trap the hairpin, we get the jump to an intermediate state, and then we get a second jump later on. And what's actually happening here is the laser tweezer is unzipping this single-stranded DNA. And so this is the intermediate step where the DNA is half as long, and this is the higher step where it's twice as, where it's the full length of 20 bases, okay? So you see an increase in transmission that eventually gets you to the same height as a single-stranded DNA that doesn't unzip, okay? So what can you use this for? Well, you can study protein-DNA interactions using this effect. So what we have here is a protein P53. Now, this is being reported to be mutated in over 75% of known cancers. This is an anti-tumor protein. And what this anti-tumor protein does is it binds to DNA. Okay, so we want to study the binding and how mutations influence this binding. So here you have this single strand of DNA that's hairpinned, and we trap this in the presence of P53, this protein, this anti-tumor protein. And what we notice is that you still get this unzipping of the single strand of DNA, but the time in the intermediate state is much longer. And the reason the time is longer is because you've created an energy barrier by binding this protein to this hairpin, to this double strand here. And so what this binding is doing is it's actually preventing the DNA from unzipping. Okay, so you can monitor and you can estimate the energy of this potential barrier that you've created for this unzipping event. Okay. But now you can add a mutant form of the P53. This is a point mutation, so it still binds. It's being shown that this still binds very well to the DNA. But what we noticed in our experiments is that the unzipping time didn't change at all. Whereas in the previous slide, we saw that the unzipping time changed by orders of magnitude on average, okay? So what we really have here is a way to probe the interactions of DNA and proteins. And now you can imagine if you could come with a drug that corrected a misfolded P53 molecule, and then you could actually see if this drug was affected by doing this experiment again and seeing if you could take this type of unzipping time trace. So this is the average time to unzip, and turn it into this one that we have here, the red curve over there, where it takes longer to unzip. Okay, so I mentioned that this noise in the trapping signal, once we've trapped a polystyrene particle, for example, it's not actually noise. It's telling us valuable things about the particle that's being trapped there. And so I'm going to talk more about that now. So one thing that we can do with this noise is figure out the size of the particle that's trapped. And these are different protein size standards. And we notice that the amplitude of the noise, okay, which is given on the vertical axis, scales linearly with the size of these globular proteins that are roughly spherical, okay? So this is quite interesting because you can use this to trap a protein and then decide based on the weight, molecular weight, what size of protein it is that you've trapped. There's an interesting data point here. There's a vertical line that essentially corresponds to trypsin and this other protein, I think it's ovalbumin, and what this trypsin does is it actually cleaves off this part of this other protein. And so this vertical line is the sum of the weights, of the molecular weights of the trypsin plus this protein. And the actual data we measure is shifted from this vertical line. And this arrow shows you how much it's shifted, and that arrow is basically the weight of this piece that's being cleaved off. So what we're able to see is we can measure, with sort of kilodalton type of accuracy, the size of protein interactions and when pieces are being cleaved off at the single molecule level. These are single particle experiments. Okay, we can also do things like antibody protein interactions. This is an example where we first trap a protein. It's bovine serum albumin again. And then we flow in a flow cell type in configuration now. We flow in an antibody that's specific to that protein, and you get a second step where you get this binding. And we did control experiments where essentially we looked at non-specific to see if there's any interactions for non-specific antibodies, and we don't see the second step over there. Okay, so this is a way to probe antibody protein interactions at the single particle level. Okay. Probably more exciting than that is the ability to actually see small molecule interactions with proteins. So this is a protein here. This is streptavidin, and streptavidin has 4 binding pockets for biotin. Okay. So when we trap streptavidin without the biotin, we see this noise But if you look closely at the noise, it actually has a relatively long time scale associated with it. There's a lot of character here. And we can do an autocorrelation of this noise signal, and we get a trace that looks like this. But now if you add biotin, all of a sudden this noise tightens up a lot, so the time scale becomes a lot shorter, and your trace of your autocorrelation looks like this. So this is a single protein molecule, and what we've been able to see is the bind-in of 4 small molecules to the single protein. It's a very distinct behavior. Okay, it's like night and day there. Okay, so what would this be useful for? Well, here's an example of an actual drug. This is aspirin. And aspirin will bind to cyclooxygenase. And if you just take this, the autocorrelation of the noise of the trap-in of a cyclooxygenase protein, then you get this signal over here. But then if you add the aspirin to it, all of a sudden it tightens up again. So you can actually look at drugs, small molecule drugs that are interacting with proteins at the single molecule level, 1 protein at a time. Okay, this is a similar experiment. It's the biotin streptavidin again, except now that we have a different type of streptavidin, we have a monovalent streptavidin, which is essentially only one of the binding pockets is active. And you see, again, the interactions with biotin and the differences between the monovalent and the regular streptavidin are all clear from these autocorrelation traces. Okay, so we can tell a lot about the proteins that we're looking at. Okay, now, the interactions I've been talking about so far have been actually quite strong interactions where a small molecule will bind, but it won't dissociate. We're also interested in sort of transient interactions where you can have association and dissociation rates. And you can do these types of experiments too. So here we have human serum albumin and a small molecule that interacts with it, and we get more light scattering or more transmission through the aperture when these two are bound together, and less when they're apart. And so we can have a discrimination line over here and do statistics on the time that the small molecule is bound and when it's unbound. And by looking at these statistics, we can get the rate constants for these associations and dissociations. Okay, so these are 2 different small molecules that both bind to human serum albumin, and we can get the molarities or the affinities of these where the binding is saturated. And so the values we get from our single molecule experiment at a single concentration are very close to the literature values. But in these literature values, what people have to do is prepare multiple different, they have to essentially do a titration. They have to do different concentrations and figure out where the saturation point is. So this is all done at a single concentration, and we can get the affinities for the binding of these small molecules with a protein. Okay. We've also been working with a drug company that's actually here, and they're interested in looking at proteins and specifically the differences between mutant and wild type proteins and how they interact. And I can't go into the details of this, but essentially we're able to distinguish looking at either the amplitude of the noise or these autocorrelation traces between the mutants and the wild types. And so this is interesting for the purposes of seeing if there are actually interactions and potentially for doing drug discovery. Okay. So now I want to go back to where I started this talk. I want to talk about vibrations of particles and how we can look at them at the single nanoparticle level. Of course, you can do conventional Raman spectroscopy on trapped nanoparticles. This is showing an example of a trap-in event for a titania nanosphere. And we just add a Raman spectrometer into our optical tweezer setup here. And we see no signal before the trap-in event, so over here in the Raman spectrum, but when we get trap-in, we see these distinctive Raman peaks which tell us that we have a titania nanosphere. Of course, you can trap multiple titania nanospheres. This happens occasionally, and at each step in the trap-in, here's a step-in event here, and here's a step-in event here. This trace is actually quite unstable, but the really sharp discontinuous steps are clearly trap-in events. And you can see an increase in the Raman spectrum. You can also trap a different type of nanoparticle like polystyrene, and you'll get a different Raman Stokes line over here. So you can identify particles based on their molecular vibrations. But actually, we're not really interested in these molecular vibrations, which tell you about the molecular constituents and their bonds, but we're interested in a larger scale type of acoustic vibration that I talked about earlier on when I opened this talk. So I'm going to talk about a technique that we've been working on called extraordinary acoustic Raman. Now you can argue about whether it's extraordinary. A lot of people like to use that word, but you can't argue that EARS is a good acronym for something where you're listening to things. So here we're looking at EARS, okay? And with EARS, essentially what you do is you have the same laser tweezer setup. It's just an inverted microscope where you shine a laser through a double nano-hole aperture. The only difference is you use two lasers. These are single frequency lasers. And you use, in this case we're using DFB lasers, and we temperature tune one of the laser's frequency with respect to the other laser. So the two lasers have different frequencies. When they interfere, this creates a fast beating signal at the difference frequency. And so this beating signal will basically modulate the amplitude of the trapping laser. And when you modulate the amplitude, you're going to be, through electrostriction, you're going to be pulling on the nanoparticle at these very high frequencies, hundreds of gigahertz type of frequencies. Okay, so we use this type of setup. When you hit a vibrational resonance of this particle, all of a sudden you heat it up, and it starts to move around more in the trap. So you see an increase in the noise. That's the thermal motion of the particle inside the trap. Okay, so this is a way to get at the vibrational motion of a nanoparticle by exciting its thermal motion. So this is showing you a sweep where we change the frequency of one of the lasers with respect to the other, and we look at the amplitude of the noise, and you see an increase when you hit a resonance of this nanoparticle. Okay, so it's a way to get the Raman spectrum of the acoustic modes of a nanoparticle without actually measuring the Raman spectrum itself. You're just measuring the change in the thermal noise or the motion of the particle in the trap. So here's an example on polystyrene nanospheres. Again, I'm showing you these L equals 2 and L equals 0 modes. And the vertical lines are what Lamb's theory would predict. And this is a sweep where we essentially change the frequency of one of the lasers, and you see these strong peaks over here. As well, we can change the size of the nanoparticle, and you see that the frequency we get matches with Lamb's theory for different sizes. So we have theory and experiment here, so there's pretty good agreement there. Just to compare this, this double peak of the L equals 2 and the L equals 0 modes with past work, a lot of people have done conventional Raman on nanoparticles, usually not just single nanoparticles, but actually large ensembles of these nanoparticles. And so they can see, for smaller nanoparticles, they can see these two peaks as well, but they're not very well resolved. And they're looking at millions of these, at least. As well, with conventional Raman, you're kind of fighting against this inelastic, the elastic scattering line, the zero loss line, or the Rayleigh line. And so this is sort of limiting the how low wave numbers you can go to. We're in the single-digit wave number regime, no problem. They can really only turn on at around 5 wave numbers or so. Of course, there have been works on microbrilliant and other types of effects. So far, the smallest particle that I've seen that has been studied with microbrilliant has been 200 nanometers. So again, I think this approach is really extraordinary compared to what's been done before. We don't have these ensemble averaging because we're looking at single nanoparticles, one at a time. Okay. So when you have this very high resolution in the wave number regime, or if you like to talk about gigahertz, well sub-wave number resolution, then what you can do is resolve things like anisotropy in your crystal. Like here's a titanium nanosphere, and because it has an anisotropic lattice, it's going to have different sound velocities along the different axes. And as a result, you're going to have one of the peaks is going to split. So you can see a very clear splitting of the ear peak, the extraordinary acoustic Raman peak. And then we can look at proteins, and there are a whole bunch of different proteins that we've looked at here. And what's very clear about these spectra is that all of them are very different. Okay? Depending on the size of the protein and the form of the protein, we get very different spectra for all of these. And we're currently working on trying to interpret these vibrational spectra. There's a lot that needs to be done in this area. And I'm giving an invited talk in the optical tweezer section, and I'll go into detail about our progress and lack thereof on this particular topic. Okay. Another thing is that this is highly reproducible. If you have multiple trapping events on the same solution of proteins or on different days or on different apertures, you get the same spectra. And what's shown here is an overlay of multiple different experiments, and the average is the thick line also shown. So you can see that we're getting the same data every single time we're trapping these proteins. Okay, so now I mentioned that I'm going to give more detail about this. I'll just quickly talk about how we could try to look at modeling of molecular vibrations, of acoustic vibrations of these proteins, and try to interpret these peaks. So this is the spectrum of conal Booman. And what we have here is 2 peaks, and then a peak, and another peak, and another peak. And you could say, well, in the spectra we observe, we do sort of see a double peak feature over here. And then we see another peak. Maybe this peak is missing. But you can try to interpret these peaks in terms of the vibrational modes that you would achieve through some sort of Hessian analysis of the molecular dynamics here. Okay, so there's this rock-in mode, there's this kind of shear mode, and there's another version of the shear mode. So these are the motions that actually allow these proteins to do various types of jobs and be useful within your body. Okay. Now we can also do these types of acoustic measurements on DNA. So with a single strand of DNA, we can trap them. I've already shown you that we can do this. But essentially, it's like a linear chain of masses on a spring. And so we can do the same type of experiment where we sweep the frequency of the one laser with respect to the other. And so we essentially sweep in the beat frequency. And this is actual data here. You see, when you're off resonance, you get a low amount of noise. This is a sort of a Gaussian distribution that's fit to this noise. And when you hit the vibrational resonance of the single strand of DNA, you get this increase in the noise. And so we can plot this RMS, the amplitude of the noise, and see this peak. Okay. So we can, for this single strand of DNA, I said we could think of this as essentially just a 1D lattice, okay, of vibrations on a 1D lattice, and there's a well-established theory for this. And so we can figure out the frequencies of a 1D lattice and match those to what we've actually obtained in the experiment. Now, of course, we can change the length of the single strand of DNA, and this will shift the frequencies of the fundamental mode of this lattice. But also you can change the sequence because the different bases all have different masses. And that will change the frequency as well. And so by doing this, you can figure out what is the sort of the mass of your single strand of DNA as well. Now, we think that by looking at multiple resonances, not just the lowest order one, and by improving the resolution of our experiment, you could perhaps actually even just do some sort of sequencing of these small fragments of DNA. I'm not sure if that would be interesting, but certainly it's interesting from a technical point of view. Okay. So we've also trapped larger particles like viruses. This is MS2. It's a phage. It's a bacteria virus. And so it's essentially a 27 nanometer protein shell. It's like a protein crystal around some water and genetic material. And when you trap this, we get a jump again. And then we did the extraordinary acoustic Raman on this, and you see these not very well-resolved peaks. And there's been a lot of interest in this in the literature, and a lot of the works in the literature have suggested that essentially you wouldn't really see very strongly resolved peaks either. But you can imagine you might be able to use this to identify different viruses and stage them. And this is all a single particle measurement. So this is one virus particle at a time. Again, I don't have time to go into the details about what we're doing on this and what's been done in the past, but I will be giving an invited talk in the optical tweezer section on Wednesday. So if you're interested in hearing more about that, you can come to that. Okay. Now this is a nanotechnology, and with any nanotechnology, you face the challenge of how do you make it highly reproducible, how do you scale it up, how do you basically how can you get to the stage of mass fabrication. So we've used this approach of template stripping, which was pioneered in the labs of Sang Hwan Oh and David Norris. And essentially this approach uses, it's kind of a replica or templating approach where you use the fact that gold does not stick at all or very well to silica. Okay, so on a silicon wafer, we can create a master pattern of these double nanoholes, and then we evaporate gold onto this, and then we apply some epoxy glue and use UV to treat it, and we can strip off the gold then, and then reuse the master template after etching out that gold over there. So in doing so, we can reuse this master many times and create nanoscale features without having to use a nanofabrication tool like the focused ion beam milling machine that we use to make these nanoholes. So we only have to use that once to create the master. So this is a way that we can cycle and with very rapidly make many samples with these double nanoholes. And the double nanoholes, the size of the gaps then can go down below 10 nanometers quite reliably with this approach, and we've shown trapping with this. Now, another thing you might want to do, though, is get rid of the entire optical microscope. If you're thinking about making this approach very scalable and benefiting from the fact that you have a nano-scale ability to trap single nanoparticles, then you want to do the whole thing on the end of an optical fiber. Okay, so you put these double nanoholes on the ends of optical fibers. Here we've etched out the core to basically identify where the optical mode is going to be, and we put the double nanohole there, and we've achieved trapping with these on the ends of fibers as well. Okay, so in that way, we could imagine an experiment where you have multiple fibers and you can probe multiple proteins in a sort of microwell plate type of setup. So you can really scale this up to large levels. Okay, so I just want to conclude my talk to sort of sum up where we are. We're looking at nanoscale objects, but specifically compared to other optical tweezer systems and what's been done in the past, we're interested in things that are in the 1 nanometer to 50 nanometer range, okay? If you're going above 100 nanometers or so, you can use conventional optical tweezers. So we're kind of really targeting this range. And I've shown you experiments involving all of these different things, protein complexes, DNA, viruses, antibodies. As well, you can imagine looking at solid state types of materials, colloidal particles, nanoparticles, carbon, nanocarbon, things like that. So I think there's a lot of potential of this approach to really probe using acoustic Raman and various other approaches with the nano tweezers to really understand these materials at the nanoscale. I should mention that the acoustic Raman it works on electrostriction, and there are other related approaches like optical Kerr effect. This is again looking at ensembles in proteins, but lots of people are interested in trying to see these vibrational resonances in a sort of terahertz or lower range. So this is a way of kind of doing terahertz spectroscopy without actually generating a terahertz wave. Okay, we just beat two lasers and we get the terahertz information about the vibrational motion of these particles. Okay, so with that, I'll thank you and I'll take any questions that you have.